Hello, in this video, we're going to be talking about translation. So let's not waste any of your time. Let's get right to it. Now, when we're talking about translation, the first thing we want to think about is the idea is that now the RNA strand is made, and we are translating that into the amino acid chain, of the polypeptide chain, and that's got some similar to transcription. There's the three main sections: the initiation, the elongation, and the termination. And of course, as a reminder, the process of expressing a gene into a protein is a two-step process: transcription, DNA to RNA, and in translation, RNA to the protein. All right, if we want to think about the steps of this working, we want to first think about all the parts that are put together initially to make it happen. So the first thing that's involved is that initial tRNA molecule, and of course, it always starts with the same amino acid, methionine, and when it says it's charged with, what it means is that it begins the process. When that one shows up, we know the process is starting, and we're ready to go. Along with that initial tRNA molecule, there's that small subunit, which is the bottom part, and then of course the mRNA strand, which is going to be red, and then something called the Kozak sequence, which isn't in your diagram, but it's simply what that is, similar to the tRNA molecule getting the methylene molecule added to it to make it more efficient and to help improve its process. It's the same thing here. This Kozak sequence is added, and it's simply added, or the function of that is to make sure that the RNA molecule is properly read and translated. And part of that process of making sure everything works right is you've got your ribosome binding sequence. So as the RNA is being positioned onto the small unit, this sequence is designed to make sure it's done correctly. Then once that's placed, then the large subunit is now added, and then the process of placing that initial tRNA onto the P site, as we saw in the last video, and then the next one onto the A site. And of course, as I mentioned, that initial amino acid typically is methionine, because that corresponds with the start codon. And so since when we looked at the codons in a previous video, there were 3 stops, but only 1 start. So typically all RNA strands start with that starter codon, and since each codon codes for a particular amino acid, then typically methionine is the first amino acid added to a strand. Okay, so after initiation, we know it's elongation, and we know from the previous video when we did transcription that initiation is a fairly short process, termination is a really short process, but that elongation is the part where all the work is being done to make that amino acid chain longer, hence the term elongation. But we're just simply talking about adding amino acids. So when we talked about in the last video that process, we talked about the P site getting the first one, the A site getting the second one, and then they shift down to the E and P, and then we keep adding amino acids into that spot, and of course then it just becomes a cycle like an assembly line, and as each amino acid is added, there's a bond between the previous one, so you get like a long strings of pearl, and of course I already mentioned that methionine is the first one. And what's on the screen here is just simply that process. And if you just follow from the top and work your way around they're kind of color-coded red and yellow, so you can see when one moves. But it's just simply an assembly line where we go from P to E, A to P, and then another one, the new one is put at A, and then we just keep moving them down one at a time. And of course, this is the chart that we saw earlier, the genetic code amino acid chart. And just a reminder that the take home here is that specific codons code for specific enzyme or amino acids, but that amino acids can be selected for by a variation or variety of codons. Some amino acids can use any or all four of the codons that start with that particular letter and have the second letter the same, or others are split where the first two letters and then the third letter is variable. But it's typically never the middle letter variable, and certainly not the first letter. That'd just be a whole different box in that case. All right, and so now we're at termination. We've read them, and we keep adding codons at the A site, and they keep moving down to the assembly line, and then we eventually get to a stop codon. And when that happens, there's transcription factors or beginning factors at the beginning. When we reach that stop codon, there's factors in place that stop the process. We are done here. And then, of course, then things start to fall away. The tRNAs will leave. The RNA strand will jump off of the small subunit and kind of break everything down. All right, and again here, just another reference point to what's happening and how all this process works out. But again, that's pretty small. But again, that's in your PowerPoint. You can see that, and you can certainly Google that as a reference point. But it does have all the steps for you listed in one spot. All right, a couple things, other things to mention to kind of clean up everything. When we're talking about codons and the genetic code, that central dogma that we talked about earlier, we want to think that, or just remind ourselves that each combination of 3 nucleotides, that's a codon. Okay, the first nucleotide in every codon is always the same, and then the variations come apart from that. And of course, a particular codon always specifies the same amino acid that never changes. So it's always a set of 3. The first letter is typically always the same, or I should say is always the same, and in each set of 3 codes for a specific amino acid. Well, that's our video over translation. When we come back next time, we're going to be looking at prokaryotic transcription. We'll see you then.